Welcome to another episode. Before we begin, take a moment to settle in, wherever you are. Maybe it's late, maybe you're resting, maybe you just want to drift into something thoughtful. I'm glad you're here. Let me know in the comments where you're listening from and what time it is there. And if you enjoy journeys like this, calm explorations of science, history, and wonder, consider subscribing. It helps this little corner of the internet stay alive. Tonight, we're going back to the very beginning. Before stories, before creatures, before even cells existed. A time when Earth itself was young, raw and wild, spinning under a newborn sun. The air was poison, the oceans boiled, meteors fell like rain. And yet, from this chaos, something unthinkable began to take shape. Something that would learn to breathe, to move, to feel, to wonder about its own beginnings. We're going to trace that story, from lifeless matter to the first spark of biology, from rock and water to you. This isn't just a tale of science, it's a meditation on existence itself, on how the atoms that make you once drifted aimlessly through space until one day they started to live. So, as the night grows quiet and your thoughts slow down, let's go back to a world before life, to where it all began. When Earth first formed, about 4.54 billion years ago, it wasn't the calm blue planet we know today. It was a world still under construction, a place of fire, metal, and collisions. The solar system itself was young. Around a newborn sun, clouds of gas and dust spiraled, clumping together under gravity. Within a few million years, those clumps became the first planetary embryos. One of them grew large enough to become Earth. Back then, temperatures on the surface easily reached 2,000 degrees Celsius, 3,600 degrees Fahrenheit, hot enough to keep most rock molten. The sky was filled with ash and vaporized metals. Asteroids bombarded the surface constantly, some the size of cities. This period, called the Hadean Eon, lasted from about 4.6 to 4 billion years ago. The name comes from Hades, the Greek underworld, and it fits. No breathable atmosphere, no oceans, and no life. Just magma fields, impact craters, and a sky choked with carbon dioxide, methane, and water vapor. Yet even in that hellish setting, the ingredients of life were gathering. When the planet finally began to cool, volcanic gases condensed into clouds. Rain fell, not for hours, but for centuries, filling enormous basins and forming the first oceans. By about 4.3 billion years ago, there may have been liquid water covering much of the surface. That means that within just a few 100 million years of Earth's formation, conditions were already shifting from fire to chemistry. It's hard to imagine how alien the atmosphere was back then. There was no oxygen to breathe. Instead, the air was thick with carbon dioxide, nitrogen, ammonia, and traces of hydrogen sulfide. If you stood there, which of course you couldn't, the pressure and heat would have crushed you instantly. Lightning flashed through dense clouds, volcanoes erupted continuously, and meteors still fell often enough to reshape entire continents. But these violent forces also provided something essential: energy. Lightning, ultraviolet radiation, and geothermal heat from the planet's interior all served as power sources that could drive chemical reactions. And with carbon, hydrogen, oxygen, and nitrogen in abundance, the basic building blocks of organic molecules, Earth had the raw materials needed for something extraordinary to happen. Scientists think these early conditions made Earth what's called a prebiotic reactor. In the oceans and on volcanic shores, countless reactions took place every second. Simple inorganic molecules collided, merged, and split apart again. Over millions of years, some of those random collisions began to create increasingly complex compounds: amino acids, fatty acids, and sugars. Where exactly this chemistry occurred remains a matter of research. Some scientists suggest it happened in shallow ponds that dried and refilled, concentrating organic molecules. Others point to volcanic hot springs or to the seafloor, where hydrothermal vents spewed mineral-rich water at temperatures above 350 degrees Celsius, 660 degrees Fahrenheit. What's clear is that early Earth was not a single environment, but a mosaic of extreme habitats. And in at least one of those environments, the line between chemistry and biology was crossed. We can still read the traces of that ancient world in the rocks today. Zircon crystals from Western Australia, dated to about 4.4 billion years old, suggest that liquid water existed remarkably early, perhaps just 150 million years old years after the planet formed. That's roughly the same amount of time that separates us from the extinction of the dinosaurs. Those zircons are tiny time capsules, proving that even in the Hadean inferno, water, and therefore stability, had already begun to appear. So by the time Earth entered the Archean Eon, around 4 billion years ago, the stage was set. The planet had oceans, continents were starting to solidify, and chemical energy was abundant. What it didn't have, yet, was life. But the chemistry was moving in that direction. At this point, the question becomes less about geology and more about definition. Because before we can understand how life began, we need to ask something deceptively simple. What exactly is life? When scientists try to define life, the first thing they realize is how slippery the word actually is. We tend to think it's obvious. You can tell when something is alive and when it's not. But the closer you look, the harder it becomes to draw that line. Biologists have spent decades trying to agree on a definition that works in every context, on Earth and beyond. The most common one centers around three core features: metabolism, replication, and evolution. A living thing takes in energy and matter from its surroundings. That's metabolism. It makes copies of itself. That's replication. And over time, those copies can change, adapt, and evolve. That's evolution. It sounds simple enough, but few things in biology fit these rules perfectly. Take a mule, for instance, a hybrid between a horse and a donkey. It's alive, but it can't reproduce. Some fungi form enormous underground networks that live for thousands of years without any clear individual boundaries. Certain bacteria enter dormant states, effectively turning off their metabolism for centuries. Are they alive during that time, or just potential life waiting to resume? The truth is, life isn't a single switch that flips on or off. It's more like a spectrum, a gradual transition from pure chemistry to something capable of sustaining itself. At the very edge of that spectrum lie things that blur the boundary entirely. Consider fire. It consumes fuel, produces energy, and spreads, even grows. In a sense, fire metabolizes, but it doesn't carry information, it doesn't evolve, and it can't pass traits to a new generation. It's dynamic, but not self-organizing in the biological sense. Or take crystals. They can form ordered structures that replicate their shape as they grow, one lattice building upon another. They even respond to environmental conditions. But there's no metabolism, no internal chemical network driving them forward. They simply follow the laws of physics. Then there are computer programs, self-replicating algorithms that copy themselves, mutate, and evolve within digital environments. They meet several of the criteria for life, yet we hesitate to call them living. Why? Because at their core, they're patterns without matter, information moving through silicon rather than cells. So what makes something truly alive? For most scientists, the answer lies in organization, the ability to maintain a structure far from equilibrium. Living systems constantly fight entropy, the natural tendency toward disorder. They do this by exchanging energy and matter with their environment, keeping themselves in a delicate state of organized instability. A rock left alone will just sit there, A cell left alone must constantly work to avoid falling apart. This ability to maintain complexity is what separates a living system from an inert one. But even that definition is not airtight. The most famous grey area in biology is the virus. Viruses are remarkable, packets of genetic material, usually DNA or RNA, wrapped in a protein shell. On their own, they're inert. They don't metabolize, they don't move, and they can't reproduce. But once they enter a host cell, everything changes. They hijack the host's machinery, using it to make countless copies of themselves. So, are viruses alive? The answer depends on how strictly you define the term. If life requires independent metabolism, then no, viruses fail that test. But if life is about information, replication, and evolution, then yes, they qualify. Viruses mutate, adapt, and evolve faster than almost any other biological entity on Earth. In fact, much of our own DNA is made up of ancient viral sequences, leftovers from infections that happened hundreds of millions of years ago. Around 8% of the human genome is viral in origin. That means part of what makes you human was once part of something that arguably wasn't even alive. This idea, that life may not have started as fully alive, is central to understanding its origins. Early Earth chemistry probably produced many systems that weren't quite alive, but weren't purely inert either. Molecules that could store energy, interact, and even replicate imperfectly. Over time, these proto-living systems may have evolved toward greater complexity, blurring the line until biology as we know it took over. There's also a philosophical side to this debate. Some thinkers argue that the word life may be less of a scientific category and more of a human convenience, a label we invented to describe certain kinds of organized chemistry that matter to us. From that perspective, life isn't a special substance, but a process, a temporary pattern of matter and energy that resists decay long enough to reproduce itself. In this sense, you could say that a cell is just a clever way for carbon to persist a little longer against entropy. Still, even if the definition is messy, there are features all living things share. They're made of cells, tiny compartments that separate internal chemistry from the outside world. They use information, stored in molecules like DNA or RNA, to guide how that chemistry unfolds. And they evolve, meaning they can change over generations in ways that favor survival. Everything else, metabolism, reproduction, behavior, stems from those principles. And once we understand that, we can start to ask a deeper question. How did such an organized, self-sustaining system first appear in a world of random molecules and chaotic energy? That question leads us directly to the next step in the story. The first serious scientific attempts to explain how life could arise naturally from non-living matter. A field of study called abiogenesis, born from experiments that tried to recreate the early conditions of Earth itself. It's where chemistry began to meet biology, and it started with a simple glass flask, a few gases, and a spark. By the early 20th century, scientists had begun to move away from the old idea that life was something mystical, something that couldn't arise without a divine spark. Instead, a new generation started asking whether life might be the inevitable result of chemistry itself. If Earth had once been lifeless, then at some point, natural processes must have bridged the gap between non-living matter and biology. That question became the foundation for a field known as abiogenesis, meaning origin from the non-living. The first real framework for this idea came from two scientists working independently in the 1920s, Alexander Oparin, a Russian biochemist, and J.B.S. Haldane, a British geneticist and philosopher. They proposed what became known as the primordial soup hypothesis, the idea that early Earth's oceans were once filled with a warm mixture of organic molecules, slowly forming through chemical reactions powered by sunlight, lightning, and volcanic heat. Oparin and Haldane imagined an atmosphere very different from today's. Instead of oxygen, which destroys delicate organic compounds, it was likely reducing, meaning rich in hydrogen, methane, CH4, ammonia, NH3, and water vapor, H2O. In such an environment, carbon could easily bond with other elements to create more complex molecules. Over time, these compounds might have accumulated in pools, lakes, and shallow seas, a global soup from which life could eventually emerge. The idea was elegant, but in the 1920s, it was entirely theoretical. No one had shown that organic molecules could form naturally under early Earth conditions. That proof would come 30 years later from a young graduate student named Stanley Miller. In 1952, at the University of Chicago, Miller, working under Nobel laureate Harold Urey, decided to test Oparin and Haldane's idea in the lab. He built a simple but ingenious device, a closed glass system with two connected flasks. One contained water, representing the ancient ocean. The other held a mixture of gases meant to simulate the early atmosphere: methane, ammonia, hydrogen, and water vapor. Electrodes produced continuous sparks between them, imitating lightning strikes. The apparatus ran for a week. When Miller analyzed the water at the end of the experiment, he found something remarkable. The liquid had turned a murky reddish-brown, and chemical analysis revealed the presence of amino acids, the building blocks of proteins, and therefore of life. It was the first time scientists had demonstrated that organic molecules could form spontaneously from simple, inorganic chemicals under plausible early Earth conditions. The experiment, published in 1953, made headlines worldwide. It suggested that the essential ingredients for life didn't need to come from somewhere else. They could form right here on a young planet, powered only by energy and time. Miller's results were quickly replicated and expanded. Later versions of the experiment produced not just amino acids, but also simple sugars, lipids, and nucleotide bases, the components of RNA and DNA. Even when scientists modified the gas mixtures to better reflect modern estimates of the early atmosphere, adding carbon dioxide, CO2, and nitrogen, N2, for example, organic molecules still appeared, though in smaller quantities. The implication was clear: prebiotic chemistry was not rare, it was robust. Today we know that the early Earth was an extraordinarily dynamic place, and that several different environments could have contributed to this molecular evolution. One likely site was the shoreline, tidal pools that repeatedly filled and evaporated under the sun's ultraviolet radiation. When water evaporates, dissolved compounds become more concentrated, allowing complex reactions that can't happen in dilute solutions. This process might have helped link amino acids into short chains called peptides, or nucleotides into the first strands of RNA. Another potential site was volcanic regions, where gases such as sulfur dioxide and hydrogen mixed in an atmosphere rich with energy. Lightning strikes were frequent, generating electrical discharges far stronger than what we see today. Each strike could heat the air to more than 30,000 degrees Celsius, 54,000 degrees Fahrenheit, temperatures sufficient to rearrange molecules and forge new chemical bonds. Over millions of years, these sparks may have acted as the planet's first molecular forges. There's also evidence that ultraviolet light from the young sun played a major role. With no ozone layer to block it, UV radiation bombarded Earth's surface, breaking apart molecules and creating highly reactive fragments that could recombine into new forms. What would be deadly to modern life might have been essential for its creation. Volcanoes, too, were crucial. They released not only gases, but also minerals and metals, catalysts that accelerate chemical reactions. Iron and nickel sulfides, for instance, are known to promote the formation of organic compounds under the right conditions. Such minerals could have accumulated on the seafloor, creating natural reaction surfaces where simple molecules assembled into more complex structures. And yet, For all this progress, there's still a gap between making amino acids and making life. The Miller-Urey experiment didn't produce living cells, only the parts that life might later use. But that's precisely what makes it important. It showed that the leap from chemistry to biology might not require anything supernatural, only enough time, energy, and stability. We now know that amino acids and other organics are not unique to Earth. They've been detected in meteorites, comets, and even interstellar dust. In 1969, for example, a meteorite fell near the Australian town of Murchison. When scientists examined it, they found over 70 different amino acids, many identical to those used in living organisms. That discovery suggested that the chemistry of life could begin anywhere in the universe where carbon, water, and energy coexist. Abiogenesis, then, might not be a singular miracle, but a universal process, one that happens naturally given the right conditions. On early Earth, it could have unfolded over tens or hundreds of millions of years, with countless false starts before something finally succeeded in sustaining itself. But we still don't know exactly where the key step occurred. Was it in a sunlit pond struck by lightning, in a volcanic spring rich with minerals, or in the darkness of the ocean floor? Each environment had the same essential ingredients: water, carbon, energy, and time, the foundations on which chemistry could become biology. The Miller-Urey experiment didn't end the mystery of life's origin, but it transformed it. It turned a philosophical question into a scientific one, and it set the stage for the next great realization, that the earliest chemistry of life might have begun not on the surface at all, but deep in the oceans, near vents where the planet's own heat powered a continuous laboratory beneath the waves. For most of the 20th century, scientists assumed life began in shallow ponds or coastal pools, warmed by sunlight and fed by lightning. But in 1977, an unexpected discovery at the bottom of the Pacific Ocean changed that view completely. A research expedition aboard the submersible Alvin was exploring the Galapagos Rift, a volcanic seam in the Earth's crust. When the cameras switched on, they revealed something that no one had ever seen before: hydrothermal vents, towering mineral chimneys, spewing black clouds of superheated water. This water was dark, not because of smoke, but because it carried dissolved metals and sulfides from deep within the crust. Temperatures at the vent openings reached around 370 degrees Celsius, about 700 degrees Fahrenheit, yet just meters away, the surrounding seawater was barely above freezing. The result was a landscape of extreme contrasts, hot and cold, lightless and yet vibrant with chemical energy. To the scientists' astonishment, these vents were surrounded by thriving ecosystems, dense colonies of giant tube worms, clams, shrimp, and bacteria, all living in complete darkness. No sunlight reached these depths, nearly 2,500 meters, 8,200 feet below the surface. The organisms survived not by photosynthesis, but by a process called chemosynthesis, deriving energy from chemical reactions rather than light. At the base of this food web were bacteria that used hydrogen sulfide, released by the vents, to power their metabolism. These microbes turned toxic gas into nutrients, feeding entire communities of life. It was a revelation. Life could exist, and even flourish, without sunlight. All it needed was energy and chemistry. This discovery reshaped how scientists thought about the origin of life. If organisms could thrive in such harsh conditions today, perhaps the earliest life on Earth began in a similar environment, powered by chemical energy instead of sunlight. That idea evolved into what's known as the hydrothermal vent hypothesis for the origin of life. Not all hydrothermal vents are the same. The ones found in 1977 were called black smokers because of the dark mineral plumes they emitted. These vents are acidic, extremely hot, and short-lived, often collapsing after a few decades. But later, in the 1980s and 1990s, scientists discovered another type, alkaline hydrothermal vents. These are gentler, cooler, and far more stable. Instead of releasing acidic, metal-rich fluids, they emit warm, mineral-laden water with a pH similar to baking soda, around 9 to 11. Temperatures hover between 40 and 90 degrees Celsius, 100 to 110 degrees Celsius, 200 degrees Fahrenheit, far less destructive to complex molecules. One of the most studied examples is the Lost City hydrothermal field, discovered in 2000 in the mid-Atlantic Ocean. It sits atop a mountain of calcium carbonate towers that can rise over 60 meters, 200 feet, high. Their porous walls form intricate networks of microscopic chambers, natural chemical reactors on a geological scale. Inside these vents, water interacts with the minerals in the ocean crust through a process called serpentinization, producing hydrogen gas, H2, and methane, CH4. These gases, combined with dissolved carbon dioxide, create energy gradients that drive complex chemical reactions similar to those used by living cells today. Those gradients are crucial. In modern biology, energy is captured by membranes, barriers that separate one chemical environment from another. Cells maintain differences in acidity, charge, and concentration across their membranes to power metabolism. Alkaline hydrothermal vents naturally create the same kind of separation. On one side of their mineral walls, the fluid is warm, alkaline, and rich in hydrogen. On the other, the ocean is cooler, more acidic, and rich in carbon dioxide. This contrast forms a natural battery, capable of generating electrical and chemical energy, exactly the kind of environment where primitive metabolism might have emerged. The mineral surfaces inside the vents could also have acted as catalysts, speeding up the formation of organic molecules. Iron, nickel, and sulfur compounds in particular are known to help assemble simple carbon-based molecules into more complex chains. In the lab, scientists have shown that these minerals can promote reactions that produce format, acetate, and even amino acids, all essential precursors to life. It's as if the vent walls were prebiotic factories, producing the first ingredients for metabolism billions of years before cells existed. What's striking is how closely some of these geochemical processes mirror the inner workings of modern organisms. Many microbes that live around vents today use a metabolic pathway called the acetyl-CoA pathway, one of the most ancient known. It uses carbon dioxide and hydrogen to produce organic molecules, just as the vent chemistry does naturally. Some researchers believe this is no coincidence, that life didn't just adapt to vents, but actually originated from them, inheriting their chemistry. To support this, scientists look at extremophiles, microorganisms that thrive in extreme environments. There are bacteria and archaea that live at temperatures above 120 degrees Celsius, about 250 degrees Fahrenheit, others that survive in acid strong enough to dissolve metal, and some that endure pressures hundreds of times greater than those at the surface. These organisms use ancient biochemical pathways, rely on minerals as catalysts, and often exist in conditions strikingly similar to early Earth's deep-sea vents. In a sense, they're living fossils, tiny, modern-day windows into the world's earliest life forms. The more scientists study them, the clearer the picture becomes. The earliest life may not have been a free-floating cell at all, but a network of chemical reactions confined within the pores of a vent structure. Over time, organic molecules might have accumulated, forming primitive membranes and genetic systems. Eventually, these systems could have detached, becoming self-contained protocells, the ancestors of all modern life. This vent hypothesis also offers a powerful explanation for one of life's greatest mysteries: how metabolism began before genes. Traditional primordial soup models struggle to explain how organized energy use could arise without enzymes or DNA. But in vents, The environment itself provides both structure and energy, an external metabolism that could have served as a stepping stone toward the internal metabolism of living cells. Even more compelling is the discovery that similar hydrothermal systems exist elsewhere in the solar system. On Enceladus, a moon of Saturn, and Europa, a moon of Jupiter, spacecraft have detected plumes of water vapor containing hydrogen, methane, and complex organic molecules. Both worlds have subsurface oceans in contact with rocky mantles, conditions nearly identical to the vent environments on early Earth. If life began in such places here, it could potentially exist there as well. So the story of life's origin may not have started in a sunlit pond at all, but in the darkness of the ocean, near vents where chemistry meets geology, where heat, minerals, and water worked together to produce complexity from chaos. These vents are reminders that life doesn't necessarily need light, warmth, or even a surface. It needs energy, chemistry, and stability. And the deep sea, once thought barren, offers all three. The more scientists learned about the complexity of life, the more one question stood out. If early Earth was filled with organic molecules, how did they organize themselves into something capable of storing information and copying itself? Because without replication, chemistry stays random. It never accumulates improvements or evolves. Life, as we know it, needed a way to remember. In modern biology, that role belongs to DNA. It carries the instructions for building every cell, every protein, every organism. But DNA doesn't act alone. It needs proteins, enzymes that read, copy, and repair it. And those proteins, in turn, are built using instructions stored in DNA. It's a closed loop. DNA makes proteins, and proteins manage DNA. So which came first? It's the classic chicken and egg problem of molecular biology. In the 1960s and 70s, researchers began to suspect that the answer might lie in a third molecule, RNA, or ribonucleic acid. RNA is often described as DNA's less stable cousin. It's made of a similar backbone of sugar and phosphate, but it uses a different sugar, ribose instead of deoxyribose, and one different base, uracil instead of thymine. But the key difference isn't just chemistry, it's function. RNA can both store information and perform tasks inside the cell. That dual ability makes it a strong candidate for the molecule that started it all. In today's cells, RNA acts as the messenger that carries DNA's instructions to the ribosomes, where proteins are made. It also plays structural and catalytic roles. It can fold into complex three-dimensional shapes that help it interact with other molecules. That versatility suggested a radical idea. Maybe there was a time, long before DNA and proteins, when RNA alone carried out the essential functions of life. This idea became known as the RNA world hypothesis. The concept gained real momentum in the 1980s after two landmark discoveries. In 1982, Thomas Tech and his team at the University of Colorado were studying how RNA molecules were processed in a single-celled organism called tetrahymena. They discovered something completely unexpected. One RNA molecule could cut and rejoin itself without any help from proteins. That same year, Sidney Altman at Yale found similar behavior in a different system, an RNA enzyme involved in processing transfer RNA. These molecules could catalyze reactions, something scientists had always assumed only proteins could do. They were named ribozymes, RNA enzymes, and they changed everything. For the first time, scientists had proof that RNA wasn't just a passive carrier of genetic information. It could also act as a biochemical machine, shaping and driving reactions on its own. This meant that an early world made entirely of RNA was not only possible, but chemically plausible. In such a world, RNA molecules could have performed both major tasks required for life. They could store information, by carrying sequences of nucleotides that could be copied, and they could catalyze reactions, including those that help make more RNA. In other words, RNA could, in theory, replicate itself. If true, this would bridge the gap between chemistry and biology. Imagine early Earth's oceans filled with countless random RNA-like strands, formed from nucleotides that accumulated in the primordial soup or around hydrothermal vents. Most of these molecules would have been inert, falling apart as quickly as they formed. But occasionally one might fold into a shape that made it more stable or better able to help nearby nucleotides join together. Such a molecule could, by pure chance, start making copies, imperfect ones, but enough for natural selection to begin its work. Replication with variation, the fundamental engine of evolution. Laboratory experiments have supported parts of this idea. In the 1990s, molecular biologists Gerald Joyce and Tracy Lincoln developed synthetic RNA systems that could assist in copying fragments of themselves. Later studies showed that small RNA molecules could evolve, literally, in the lab, changing sequence over successive generations to become more efficient catalysts. It wasn't full self-replication, but it was close. We also see hints of this ancient RNA world still operating inside modern cells. The core of the ribosome, the structure that builds proteins, is made not of proteins, but of ribosomal RNA, rRNA. It's the RNA itself that performs the key reaction linking amino acids together, not the surrounding proteins. That discovery, made in the late 1990s, confirmed that the most fundamental process in biology, making proteins, is driven by a molecule that likely predates proteins altogether. Every living organism on Earth, from bacteria to humans, carries this molecular relic from the RNA age within its cells. But the RNA world hypothesis, while elegant, still faces challenges. For one, RNA is chemically fragile. It breaks down quickly in water, especially at high temperatures. Its sugar, ribose, is difficult to form and even harder to stabilize. This raises the question, how could enough RNA have formed and persisted on the early Earth for life to take hold? Some researchers suggest that before pure RNA, there might have been a simpler system, molecules that combined parts of RNA with other backbones, or even a mix of different nucleic acids. Others propose that mineral surfaces, like clay or iron-sulfur compounds, could have acted as scaffolds, helping RNA strands assemble and stay intact. Laboratory experiments have shown that montmorillonite clay, for example, can help link nucleotides into short RNA chains under plausible early Earth conditions. These findings hint that the first steps toward an RNA world could have taken place on solid surfaces, not in open water. There's also the question of energy. RNA replication requires activation. Molecules need to be in a high-energy state to form the necessary bonds. Natural sources of such energy could include ultraviolet light, volcanic heat, or chemical gradients around hydrothermal vents. Some researchers even propose that early RNA molecules might have exploited those same gradients, using natural proton flows across mineral membranes, much like modern cells use across lipid membranes today. So, while no one has yet created a fully self-replicating RNA system in the lab, the evidence keeps pointing in the same direction. RNA, or something very much like it, was the central player in life's first steps toward complexity. From this perspective, the evolution of DNA and proteins looks less mysterious. Once RNA-based systems began to thrive, DNA could have evolved as a more stable data archive, better suited for long-term storage. Proteins, meanwhile, are more versatile catalysts, faster, stronger, and more specialized than RNA. Eventually, these newer molecules took over most of RNA's roles, leaving it as the messenger and coordinator we see today. But RNA's fingerprints are still everywhere, in every cell, every translation of a gene, every molecule of life. In a sense, all of biology is still the echo of that early RNA world, a world where information and chemistry were one and the same. And yet, even if RNA was the key, we still haven't answered one crucial question. How did those first molecules become enclosed, protected, and organized into something we could recognize as a living cell? That next transformation, from molecular chaos to biological order, would mark the true dawn of life as we know it. At some point on the early Earth, chemistry began to organize itself. Amino acids were forming, sugars and nucleotides too. Energy sources were everywhere, heat from volcanoes, lightning, and the chemical gradients around hydrothermal vents. But there was still a problem. Even if complex molecules like RNA could replicate, they were drifting freely in the ocean, constantly diluted, broken apart, or destroyed by environmental change. For life to truly begin, those fragile systems needed protection, a way to create an inside and an outside. That boundary would come in the form of something simple but revolutionary: the cell membrane. Among the molecules forming on early Earth were lipids, long chains of carbon and hydrogen, with one end that loves water, hydrophilic, and one that hates it, hydrophobic. That dual nature gives lipids a special property. When placed in water, they spontaneously arrange themselves to minimize contact between their water-fearing tails and the surrounding liquid. In the right conditions, these molecules organize into bilayers, two layers of lipids with their hydrophobic tails facing inward and their hydrophilic heads facing outward. The result is a microscopic bubble known as a vesicle. This process doesn't require enzymes, energy, or any special conditions. It happens entirely through self-assembly, driven by simple physical forces. Even in the lab, scientists can create vesicles just by shaking fatty acids in warm water. Within minutes, they form spherical membranes under the microscope, tiny structures that behave astonishingly like primitive cells. In the 1990s, biophysicist Jack Szostak and his colleagues at Harvard began studying these primitive structures, calling them protocells. They found that certain fatty acids, especially those with 10 to 12 carbon atoms, form stable vesicles at moderate temperatures, around 40 degrees Celsius, 104 degrees Fahrenheit. These vesicles can grow by absorbing more fatty acids from their surroundings, and can even divide under physical stress, like agitation or mild heat. In other words, they show some of the same behaviors as living cells, despite having no genes or proteins at all. This self-assembly had a profound consequence: compartmentalization. Once a lipid bubble forms, it traps a bit of the surrounding water and whatever molecules happen to be in it. That creates an internal environment, separate from the external world, where chemistry can unfold in a more controlled way. In open water, molecules are constantly dispersed, but inside a vesicle, they're confined, increasing the chance that reactions occur and sustain one another. It's like the difference between trying to start a fire in a rainstorm versus inside a sheltered cave. The boundaries make all the difference. With compartmentalization came the birth of individuality. Each vesicle was a unique microenvironment containing a slightly different mixture of molecules. Some might have had compounds that helped them grow faster. Others might have included fragments of RNA or amino acids that interacted in new ways. This diversity created a simple form of natural selection. Protocells that were more stable or efficient at taking in new materials survived longer than others. From this gradual filtering, the first pre-biological evolution began. But membranes did more than just contain chemistry. They also allowed stability. A lipid bilayer is semi-permeable. It lets water and small neutral molecules pass, but keeps larger or charged molecules out. This selective barrier meant protocells could hold onto valuable molecules like nucleotides or amino acids while maintaining concentration gradients. In modern cells, such gradients are used to generate energy through the movement of ions across membranes, a process that powers nearly all life. In protocells, the same basic principle may have existed in a primitive form. Differences in acidity or chemical concentration between the inside and outside could create potential energy that drove simple reactions. For example, in alkaline hydrothermal vent environments, the external seawater was acidic compared to the alkaline vent fluids. If protocells formed in these regions, their membranes could naturally maintain pH gradients, harnessing the same kind of chemical energy used in modern metabolism. This may have helped early protocells sustain simple reaction cycles long before the evolution of enzymes or complex metabolic networks. Over time, certain molecules trapped inside protocells could have stabilized the membranes themselves. For instance, peptides, short chains of amino acids, can insert into membranes and strengthen them, while also influencing permeability. Some researchers have proposed that peptides and lipids co-evolved, each supporting the other's stability and functionality. The next great step came when RNA, those fragile information-carrying molecules from the previous stage, became enclosed within these protocells. This marriage of information and compartmentalization marked the beginning of the first true living systems. Experiments by Jack Szostak's group have shown that if you mix fatty acid vesicles with RNA strands under the right conditions, some vesicles will spontaneously trap the RNA inside as they form. Even more interestingly, the vesicles don't block small molecules from entering, meaning new nucleotides can diffuse in, potentially feeding replication reactions. When scientists supplied the system with activated nucleotides, they found that RNA copying reactions could occur within the protocells, protected from the outside environment. The vesicle provided both shelter and concentration, allowing reactions that would otherwise be too slow or too easily disrupted. This combination of RNA replication and membrane dynamics creates a kind of minimal life cycle. Vesicles grow as they take in fatty acids, divide under gentle heat or agitation, and pass on some of their internal contents, including RNA strands, to their offspring. It's not conscious, not coordinated, but it's the beginning of inheritance, the first glimmer of biological continuity. In computer simulations and lab experiments, this kind of system can even show competition. Protocells that contain RNA or catalytic molecules that help them grow tend to outcompete others for resources. Those without such helpers gradually vanish. In this way, chemistry begins to obey the rules of evolution, variation, inheritance, and selection. One of the key challenges early protocells faced was maintaining osmotic balance, the pressure created when solute concentrations differ inside and outside the membrane. If too much material accumulated inside, the vesicle could burst, but some fatty acids naturally swap between the inner and outer layers, adjusting membrane tension and allowing stability across changes in concentration. That built-in adaptability made them surprisingly robust. Another important discovery was that membrane growth could be linked to internal chemistry. When RNA replication occurred inside a vesicle, it changed the internal conditions, often making the protocell more acidic. That acidity drew more fatty acids from the environment into the membrane, making it grow. As a result, protocells with active chemistry inside literally grew faster than inactive ones. It's a primitive but powerful feedback loop, metabolism driving growth. When growth exceeded a certain threshold, the vesicle would divide, distributing its contents roughly equally. Each new protocell inherited a portion of the internal molecules, including any catalytic RNA. This simple process resembles cell division in principle. Though it happened entirely through physical forces, not biological machinery. Over time, protocells may have developed more sophisticated chemistry. Fatty acid membranes could incorporate other molecules, such as glycerol phosphates, making them sturdier and less permeable. RNA molecules might have begun catalyzing reactions that produced useful lipids or energy sources. Each improvement made the system more stable and self-sufficient. Eventually, some protocells could have evolved the ability to synthesize their own building blocks, amino acids, nucleotides, and simple sugars, from raw materials. At that point, the line between chemistry and life was crossed. No longer dependent solely on their surroundings, these protocells could control their internal chemistry, the first true metabolism. This is what scientists sometimes call the emergence of autopoiesis, self-creation, a system that maintains and reproduces itself through internal processes. In essence, the first organism wasn't a cell in the modern sense, but a self-sustaining network enclosed in a boundary. Modern biochemistry still carries echoes of this ancient transition. All living cells are surrounded by lipid membranes, and all rely on the same basic principles of energy gradients and selective permeability. Even the shapes of modern cell membranes, flexible, self-healing, and capable of fusion and division, are direct descendants of those early fatty acid vesicles. In 2019, researchers at the University of Tokyo recreated protocell-like systems using simple fatty acids and RNA fragments. These artificial cells were able to grow, divide, and maintain internal reactions autonomously for several cycles. While they were not alive in any full sense, they offered a glimpse of how simple molecules can give rise to lifelike behavior purely through physics and chemistry. When we look at modern single-celled organisms, bacteria or archaea, we can still trace their ancestry back to that era. Their membranes, their internal chemistry, even their genetic organization all carry signatures of a time when the boundary between life and non-life was blurred. The emergence of membranes completed the essential triad of life's foundation: information, RNA, metabolism, chemical energy cycles, and compartmentalization, lipid boundaries. Once those three came together, the rest was evolution. Millions of years of refinement that led from protocells to the first true biological ancestor of everything alive today. That ancestor is known as LUCA, the last universal common ancestor, the organism from which every cell on Earth, from bacteria to blue whales, ultimately descends. But LUCA wasn't the first living thing. It was the one that survived. Every living organism on Earth, every tree, every bacterium, every whale, and every human being shares a common lineage. No matter how different we appear on the surface, our biochemistry tells a single story. All life descends from a single ancestral population that lived billions of years ago. Scientists call it LUCA, the last universal common ancestor. It's important to understand that LUCA was not the first life. Countless primitive organisms came before it. Many likely perished without leaving descendants. LUCA was simply the one whose lineage endured, the genetic survivor from which every modern species ultimately evolved. In a sense, LUCA is not the origin of life, but the last common point where all the branches of life's tree converge before diverging into bacteria, archaea, and eventually everything else. LUCA lived roughly 3.8 to 4.1 billion years ago, during the late Archean Eon, a time when Earth was still geologically active, its oceans hot, and its atmosphere devoid of oxygen. The planet was far from peaceful. Meteorites still struck occasionally, volcanoes erupted continuously, and ultraviolet light bombarded the surface through a thin atmosphere. But deep below, near the warmth of hydrothermal vents, life had already taken hold. Because LUCA left no fossils, scientists have had to reconstruct its nature from a different source, genomics. By comparing the DNA of modern organisms, researchers can identify genes and proteins that are shared by every form of life on Earth. Those shared features are the ones that LUCA most likely possessed. When scientists first attempted this genetic reconstruction in the 1990s, they expected to find only a few dozen common genes. Instead, they found hundreds. As more genomes were sequenced, from bacteria, archaea, and eukaryotes, a consistent pattern emerged. LUCA wasn't a simple molecule or a half-living chemical network. It was already a cellular organism with a surprisingly sophisticated biochemistry. From these shared genetic signatures, we can outline a portrait of LUCA's life. It likely had a cell membrane built from fatty molecules that enclosed its internal chemistry. Inside, it carried ribosomes, molecular factories for building proteins. It used RNA and DNA for information storage and transfer, and it read that information using the same genetic code that all organisms use today. That code, the mapping of nucleotide triplets to amino acids, appears universal because it was fixed in LUCA's time and never changed afterward. In short, every living thing still speaks LUCA's biochemical language. Its metabolism, however, was unlike that of most organisms alive today. Genetic studies show that LUCA lacked enzymes for dealing with oxygen, meaning it lived in a strictly anaerobic environment. It probably obtained energy through chemical reactions involving hydrogen, H2, carbon dioxide, CO2, and metal ions, reactions similar to those found around modern hydrothermal vents. In particular, LUCA's enzymes suggest it relied on a pathway known as the Wood-Ljungdahl pathway, one of the most ancient metabolic routes known. This pathway converts carbon dioxide and hydrogen into organic compounds, such as acetyl-CoA, a crucial molecule still used in every cell's metabolism today. The key to this chemistry is that it doesn't require sunlight or oxygen. It's driven purely by geochemistry, by the same kinds of redox reactions that happen naturally where hot, mineral-rich fluids meet cooler ocean water. That makes LUCA's world look strikingly similar to what scientists have seen at alkaline hydrothermal vents, like the Lost City Field in the Atlantic. The environmental clues and the genetic evidence point in the same direction. LUCA likely lived deep under the sea, not in a warm pond or tide pool, but in the labyrinthine structures of a vent system, where heat and minerals sustained a steady source of energy. LUCA's genome, reconstructed from modern comparisons, contained genes for enzymes that bound iron, nickel, and sulfur, elements abundant in vent minerals. It also included components of ATP synthase, the rotary enzyme that nearly all life uses to produce energy. This enzyme works like a tiny molecular turbine, spinning as protons flow through it across a membrane. The fact that LUCA had parts of this complex machinery suggests that it already exploited proton gradients, natural differences in charge and acidity between inside and outside, to power its metabolism. In other words, LUCA was using a primitive form of chemiosmosis, the same principle that drives your own mitochondria today. That continuity is extraordinary. The same basic energy mechanism that powers human cells was already in use nearly 4 billion years ago. However, LUCA didn't possess everything we might associate with life. It probably lacked the full enzymatic toolkit needed to synthesize certain amino acids, nucleotides, and lipids. That means it may have depended partly on its environment, on geochemical reactions producing some of its raw materials. It was, in many ways, half biological and half geological, a hybrid that lived on the boundary between Earth's chemistry and biology. The temperature of LUCA's habitat was probably moderate by vent standards, around 70 to 90 degrees Celsius, 160 to 195 degrees Fahrenheit, hot but not boiling. Stable, mineral-rich, and shielded from ultraviolet radiation, this environment offered everything early life would need: energy, protection, and raw materials. The presence of hydrogen gas and carbon dioxide in abundance made vent systems natural energy sources for carbon fixation, converting inorganic carbon into organic molecules. Modern analogues of this lifestyle still exist. Some of the oldest and most primitive organisms known, such as methanogens and acetogens, use similar metabolisms to what LUCA likely used. They combine hydrogen and carbon dioxide to form methane, CH4, or acetate, CH3COOH, releasing energy in the process. These microbes thrive today in hydrothermal vents, hot springs, and even the guts of animals, living fossils of a metabolic world that has never truly disappeared. From LUCA's genetics, we also learn about its structure. It was most likely unicellular, roughly bacterial in scale, perhaps a few micrometers across. Its DNA was probably circular, floating freely inside the cell, much like modern bacteria and archaea. It used ribozymes and metal cofactors to catalyze reactions, relying heavily on transition metals such as iron and nickel, which were plentiful in the early ocean. It's also clear that LUCA lived before the great split between the two main domains of life, bacteria and archaea. Those two groups diverged from LUCA's lineage early on, developing distinct membranes and molecular machinery. Bacteria went on to populate nearly every environment on Earth, while archaea thrived in more extreme conditions: high temperatures, acidity, salinity. Much later, one lineage of archaea merged with a bacterium, giving rise to the first eukaryotic cells, the ancestors of all plants and animals. That makes LUCA, indirectly, the ancestor of both your body and the microbes in your gut. What's most remarkable is how much of LUCA's biology remains with us. The structure of DNA and RNA, the universal genetic code, the use of 20 amino acids in proteins, the reliance on ATP as an energy currency, all of these features were already established in LUCA's time. Evolution has refined them, but it hasn't replaced them. That stability over billions of years shows that LUCA's design was not only functional, but close to optimal. Once evolution found that combination of molecules and reactions, there was no reason to start over. In a way, LUCA represents the moment when life became locked in, when chemistry settled into a system robust enough to endure for the rest of Earth's history. Before LUCA, life might have been messy, experimental, full of short-lived molecular variants. After LUCA, life had a clear identity: cellular, genetic, and self-replicating. It's humbling to realize how much connects us to that ancient cell. When you breathe, the process that releases energy in your mitochondria is a direct descendant of the same hydrogen-based reactions that LUCA used. When your cells make proteins, they follow the same three-letter genetic code that LUCA established. When DNA copies itself, it uses enzymes whose distant ancestors worked inside that primordial organism. On a biochemical level, you and LUCA are speaking the same language, separated by nearly 4 billion years of evolution. Yet LUCA's world was fragile. Earth then was not stable. Oceans boiled near volcanic ridges. The atmosphere was filled with carbon dioxide and methane, and asteroids still occasionally struck. LUCA's descendants survived because they lived deep underwater, shielded from radiation and cosmic impacts. That refuge may have been essential for the continuity of life during what geologists call the Late Heavy Bombardment, a period around 4 billion years ago, when impacts could have sterilized much of the planet's surface. Hydrothermal systems beneath the ocean may have been the safe zones where life persisted. LUCA's story also provides a guide for searching for life elsewhere. If life arose in hydrothermal environments here, then icy moons like Europa and Enceladus, with their subsurface oceans and vent-like chemistry, may offer similar conditions. The same geochemical principles, hydrogen, carbon dioxide, and mineral energy, could, in theory, produce life beyond Earth. When we explore those worlds, we're not just looking for aliens. We're looking for echoes of LUCA's world, perhaps another version of that same fundamental chemistry. So, when you look at a cell under a microscope, or think about your own body made of trillions of them, remember that all of it, every structure, every process, traces back to one ancient lineage that began in the dark, mineral caverns of the early ocean. LUCA connects every living thing on Earth through an unbroken chain of inheritance, stretching across eons. We'll never know exactly what that first cell looked like, or what color the vents around it glowed. But through the molecules inside us, LUCA still speaks, quietly, persistently, through every living being that carries its memory forward. Life, at its core, is LUCA's story, continuing to unfold. The story of life's beginning has always invited another question, one that reaches beyond our planet. If the chemistry of life could arise here, could it also have come from somewhere else? For as long as scientists have studied the origin of life, some have wondered whether the first living material on Earth might not have formed here at all, but instead arrived. That idea is known as panspermia, from the Greek for seeds everywhere. It's the notion that the building blocks of life, or even microscopic organisms themselves, might travel through space, carried by comets, asteroids, or cosmic dust, and seed life on any planet with the right conditions. The idea isn't new. It appeared as early as the 5th century BCE with Anaxagoras, a Greek philosopher who suggested that life exists throughout the universe and simply takes root wherever it finds nourishment. The theory re-emerged in the 19th century, when scientists like Hermann Richter and later Lord Kelvin and Hermann von Helmholtz proposed that life could be transferred between worlds via meteorites. At the time, the concept was partly philosophical, an attempt to avoid confronting the difficult question of how life might start from non-living matter. But as science advanced, evidence began to make panspermia seem less like fantasy and more like a possibility worth testing. The first clue came not from biology, but from astronomy. Throughout the 20th century, spectroscopy, the analysis of light from distant objects, revealed that many molecules floating in interstellar space are organic. By the 1960s, astronomers had detected simple carbon compounds like formaldehyde, methane, and even amino acids in cosmic dust clouds. It was becoming clear that organic chemistry is not rare. It's widespread across the galaxy. If those compounds could form naturally in the vacuum of space, perhaps they could also survive long enough to be delivered to young planets like Earth. Then, in 1969, something extraordinary fell from the sky. A meteorite landed near the small town of Murchison in southeastern Australia. When scientists examined the fragments, they found a treasure trove of organic material. Inside were over 70 amino acids, including 19 found in living organisms, along with sugars, nucleobases, and a variety of hydrocarbons. Many of these compounds showed both left and right-handed molecular forms, a sign that they were not products of terrestrial biology, but rather formed through chemical processes in space. The Murchison meteorite provided the first hard evidence that complex organic molecules could be synthesized in the interstellar environment and survive their journey to Earth. Subsequent analyses of other carbon-rich meteorites, such as the Tagish Lake and Orgay meteorites, confirmed similar findings. Comets, too, were found to contain organic compounds, including formaldehyde, glycine, and other amino acids, detected directly by spacecraft missions like Rosetta in 2014, which studied comet 67P/Churyumov-Gerasimenko. All of this reinforced the idea that early Earth was not an isolated chemistry lab. It was constantly bombarded by comets and meteorites carrying organic cargo, potentially trillions of tons over hundreds of millions of years. These impacts could have delivered not just water, but also the chemical ingredients for life's emergence. This version of panspermia, the weak form often called organic delivery, doesn't claim that life itself came from space. It simply suggests that space helped supply the ingredients. In that sense, it works hand in hand with abiogenesis. If the building blocks were raining down from space, Earth's early chemistry would have had a massive head start. But a more radical idea emerged in the 20th century, that not only the ingredients but life itself might have traveled here from elsewhere. In the early 1900s, the Swedish chemist Svante Arrhenius proposed a version of panspermia in which microbial spores could drift between planets, carried by light pressure from stars. Arrhenius calculated that microscopic life could, in theory, survive interstellar travel if it were shielded from radiation and cold. At first, this seemed far-fetched. Space is filled with deadly ultraviolet and cosmic radiation, vacuum, and temperature extremes. But decades later, laboratory experiments showed that some organisms, particularly bacterial spores and certain microbes known as extremophiles, are astonishingly resilient. For example, bacteria such as Deinococcus radiodurans can withstand doses of radiation thousands of times higher than what would kill a human. Others can survive freezing, dehydration, and even the vacuum of space. In 2019, experiments aboard the International Space Station confirmed that some bacterial spores could survive for years in low Earth orbit, partially shielded from ultraviolet radiation. And in 2020, the Japanese Tanpopo mission showed that clusters of microbes might survive even longer, potentially long enough to travel between planets within the same solar system. Such findings suggest that life, if it already existed somewhere, could indeed spread, at least within a single planetary neighborhood. For example, a powerful impact on Mars could eject rocks containing microbes into space, and some of those rocks might eventually land on Earth. In fact, Martian meteorites have been found here, identified by their gas composition matching the Martian atmosphere. If microbes ever lived on Mars billions of years ago, it's conceivable that their descendants could have hitchhiked across space. That possibility, known as lithopanspermia, is scientifically plausible, though not yet proven. Computer simulations show that rocks blasted from Mars could reach Earth in a few million years, and microorganisms inside them might remain insulated from radiation and heat during the journey. We don't yet know whether any actually did, but it remains an open and testable hypothesis. The most controversial form of panspermia, though, goes a step further: directed panspermia. In 1973, Nobel Prize-winning molecular biologist Francis Crick, co-discoverer of the structure of DNA, published a paper suggesting that life on Earth might have been deliberately seeded by an advanced civilization. He proposed that microorganisms or spores could have been launched aboard spacecraft, intentionally sent to new worlds to propagate life. Crick didn't claim this was true, but he argued that it was worth considering given the improbability of life's spontaneous origin under known conditions. Directed panspermia sits at the border of science and speculation. It doesn't solve the problem of how life began. It just relocates it, suggesting that it started elsewhere and was transported here. Crick himself later admitted that while the idea was provocative, it offered no real explanatory advantage. It simply moved the mystery back a step. That's the main criticism of panspermia in general. It doesn't answer the fundamental question. If life came from space, where did that life come from? Whether it began on Earth, Mars, or a distant exoplanet, we still face the same challenge, explaining how non-living matter first became alive. Nevertheless, panspermia has value because it expands our sense of possibility. It reminds us that life might not be unique to Earth, and that the chemistry of biology could be universal. In recent years, data from telescopes and spacecraft have shown that amino acids, nucleobases, and even simple sugars are found in places far beyond our planet. They've been detected in interstellar clouds, comet tails, and even Titan's atmosphere, where methane rains onto hydrocarbon lakes. In 2022, scientists studying samples from the asteroid Ryugu, returned by Japan's Hayabusa 2 mission, confirmed the presence of uracil, one of RNA's bases, and other nitrogen-containing molecules. These findings strengthen the idea that the ingredients of life are common in the cosmos. It seems that nature keeps producing them wherever the right elements and energy sources exist. So perhaps panspermia and abiogenesis aren't competing ideas, but complementary ones. Space might have provided the starter kit, while Earth, with its oceans, minerals, and energy, was the laboratory where those materials assembled into something living. It's also possible that life began in more than one place. Maybe it started here and elsewhere, independently, following the same chemical pathways because the laws of physics and chemistry guide matter toward complexity wherever conditions allow. If that's true, then the universe might be full of LUCAs, distinct ancestors of life, each born in its own planetary cradle. Still, panspermia remains an open question. We have meteorites that carry amino acids, microbes that can survive in space, and chemical evidence from across the solar system. But we don't yet have proof that a living organism has ever made the trip between worlds. Until that day comes, abiogenesis, the emergence of life from Earth's own chemistry, remains the leading explanation. It's supported by experiment, by geology, and by the principle that complexity can arise naturally when given enough time and energy. Panspermia, in that sense, is not a replacement theory, but a reminder of humility. It asks us to consider that life's story might be even broader than our planet, that the same cosmic dust that formed stars, planets, and oceans might also carry the potential for biology wherever it lands. But whether life began here or arrived from afar, one truth remains constant. It is all made of the same matter. The carbon in your body was forged in stars. The amino acids that form your proteins can be found drifting between galaxies. Life may have begun in a puddle, or in a vent, or inside a meteorite, but its ingredients were always cosmic. So in a sense, panspermia is both wrong and right. It doesn't explain the origin of life, but it reminds us that life's ingredients have always been part of the universe's natural chemistry. The seeds were indeed everywhere. Earth was simply the place where they took root. For most of human history, the origin of life was beyond the reach of experiment. It was a mystery, confined to philosophy and imagination, an event too ancient, too improbable to ever witness or test. But over the past century, that has changed. Step by step, scientists have begun to recreate the conditions of early Earth in laboratories, testing theories that once sounded almost mythical. Each new experiment doesn't just answer questions. It raises better ones, slowly tracing the line between chemistry and biology. The story begins, as it often does, with the Miller-Urey experiment of 1952, the first proof that organic molecules can form spontaneously under plausible prebiotic conditions. But since then, researchers have gone far beyond amino acids in a flask. They've started building systems that behave in limited ways like living things. One of the most influential figures in this new era of origin-of-life research is Jack Szostak, a Nobel Prize-winning biochemist whose work focuses on the creation of protocells, artificial, cell-like structures capable of growth, division, and even primitive information storage. In the 1990s and early 2000s, Szostak and his colleagues began experimenting with fatty acids, RNA strands, and simple chemical gradients, trying to find combinations that could replicate the steps from chemistry to life. What they found was extraordinary. When fatty acids are mixed in warm, slightly alkaline water, they spontaneously form spherical membranes, vesicles, that can trap other molecules inside. Shostak's team showed that these vesicles could incorporate RNA molecules, and that under the right conditions, those RNA strands could begin to copy themselves. Even more surprising, the replication process inside the vesicles caused subtle chemical changes that made the membranes grow faster. In essence, the internal chemistry promoted the survival of its own container, a simple feedback loop, one of the earliest signs of lifelike behavior. When these protocells became too large, physical forces caused them to divide, distributing their contents between daughter vesicles. It wasn't evolution in the biological sense, but it was the same logic. Systems that worked tended to persist, while those that didn't faded away. By 2013, Shostak's lab had demonstrated protocells that could undergo multiple growth division cycles, each time preserving some of their internal molecules. In a purely chemical world, these were the first hints of something that could inherit. At around the same time, David Deamer, a biochemist at the University of California, Santa Cruz, was exploring how cell membranes could have formed naturally on the early Earth. Deamer studied amphiphilic molecules, compounds with both water-loving and water-repelling parts, extracted from meteorites like the Murchison sample. He found that even extraterrestrial organics, when placed in water, could spontaneously form membranes. This meant that the first protocells might not have needed to invent their boundaries. They could have been provided by chemistry itself, delivered by meteorites, or synthesized in volcanic pools. Deamer also conducted experiments simulating drying and wetting cycles in volcanic ponds. When solutions of nucleotides dried on hot mineral surfaces, the heat and evaporation drove them to form long chains, primitive RNA strands. When water returned, the molecules rehydrated and dispersed again. Repeated over thousands of cycles, these processes could have built the polymers necessary for genetic storage. It's a striking image. Life's first molecules forming not in a single flash of creation, but through patient, repetitive chemistry driven by the rhythms of the planet. Other research groups have focused on the self-replicating behavior of RNA. In the 1990s, Gerald Joyce at the Scripps Research Institute developed synthetic RNA enzymes that could copy small fragments of themselves. His later experiments went further. In 2009, Joyce and his colleague Tracy Lincoln created two RNA molecules that could catalyze each other's synthesis, a cross-replicating pair. Given the right building blocks, these RNAs reproduced indefinitely in the lab. They even competed for resources, showing Darwinian evolution on a molecular scale. More recently, scientists have used advanced sequencing and automation to evolve RNA populations in real time. They start with random RNA sequences, then repeatedly select for those that fold into useful catalytic shapes. After many generations, sometimes just a few weeks, entirely new RNA enzymes emerge, performing functions never seen before. These experiments prove that natural selection doesn't require biology. It can happen with molecules alone, given variation and persistence. Parallel progress has come from geochemistry, from scientists who study how Earth's environment might have powered these reactions. One focus is the simulation of early atmospheres and oceans. In the decades since Miller and Urey, researchers have refined our understanding of what gases were present on the young Earth. Rather than being strongly reducing, the atmosphere was probably rich in carbon dioxide, nitrogen, and small amounts of methane and hydrogen. To test how organic molecules might form under these more neutral conditions, modern teams use plasma discharges, ultraviolet lamps, and high-pressure reactors that replicate ancient volcanic outgassing and lightning storms. Even under these milder conditions, they still observe the formation of amino acids, lipids, and nucleobases, suggesting that life's ingredients are even more resilient than once thought. In Japan, scientists at the Tokyo Institute of Technology have simulated deep-sea vent conditions, mixing hot, mineral-rich fluids with colder ocean water. Within minutes, they observed the spontaneous formation of organic compounds and even short peptide chains, the building blocks of proteins. Other experiments have shown that iron-sulfur minerals, abundant in early seafloor rocks, can catalyze the conversion of carbon dioxide into small organic molecules like formate and acetate, reproducing parts of the same metabolic pathways used by the earliest microbes. One of the leading voices in this field, William Martin, has argued that life's chemistry is essentially an extension of geochemistry, that metabolism began as a natural product of the Earth's interaction with water and rock. His work on metabolic reconstruction suggests that the chemical networks inside cells today mirror those that occurred spontaneously at hydrothermal vents billions of years ago. The enzymes we now use to process energy may have evolved from inorganic catalysts, metal clusters of iron and nickel embedded in mineral matrices. The frontier of origin of life research has also moved towards synthetic biology, building living systems from scratch. In 2021, a team at the J. Craig Venter Institute announced the creation of JCVY-SYN 3.0, a minimal bacterial cell with just 473 genes, the smallest genome known to sustain life. By stripping away every non-essential function, scientists can see which genes truly define a living system. It's an approach that, in reverse, also helps us imagine what the simplest early cells, LUCA's ancestors, might have needed to survive. Another branch of research focuses on mineral catalysis and peptide formation, two processes thought to precede the genetic era. In 2023, chemists demonstrated that small peptides could form spontaneously when carbonyl sulfide, a volcanic gas common on early Earth, reacts with amino acids under mild conditions. The reaction occurs at room temperature and requires no enzymes or advanced chemistry, producing short protein-like chains that can fold and even catalyze other reactions. This discovery suggests that peptides, like lipids and nucleic acids, might have been able to arise independently, later joining forces with RNA in the first protobiological systems. Meanwhile, computational chemists and artificial intelligence systems are now being used to map entire prebiotic reaction networks, identifying which molecular combinations could occur naturally and how likely they are to lead to lifelike behavior. These simulations explore thousands of possible reaction pathways, providing insight into how complexity can emerge from randomness. In the lab, researchers have also built microfluidic environments, tiny chips that simulate the dynamic conditions of ancient environments like wet-dry cycles, mineral surfaces, and fluctuating temperatures. By controlling these conditions precisely, scientists can observe how molecules self-organize, aggregate, and sometimes transition into forms that behave almost alive. Some of these systems even show primitive metabolic feedback loops, in which a chemical reaction modifies its surroundings in a way that promotes its own continuation, a core feature of biological systems. Together, these experiments have transformed origin of life studies from speculation into a vibrant empirical field. We can now grow protocells, evolve RNA, synthesize peptides, and recreate prebiotic reactions under conditions that match those of early Earth. Each result fills in another fragment of the puzzle, suggesting that the boundary between non-living and living is thinner than once believed. The emerging view is that life didn't begin in a single miraculous event, but through a continuum of chemical evolution, a gradual shift from random reactions to organized systems capable of self-maintenance and replication. It was not a spark, but a slow ignition, one that likely took millions of years of molecular trial and error before settling into stability. And yet, even with all this progress, one mystery remains unsolved. No experiment has yet produced a system that can fully replicate, evolve, and sustain itself indefinitely without human intervention. We can make lifelike chemistry, but not yet life itself. That elusive step, the transition from chemistry that merely reacts to chemistry that persists, remains one of science's greatest challenges. Still, every discovery brings us closer to understanding not only how life began here, but how it might begin elsewhere. The same experiments that recreate ancient Earth also hint at what could happen on Mars, on Europa, or in the oceans beneath the ice of Enceladus. The question of life's origin is no longer confined to the past. It's a guide to the future of exploration. For now, we continue to chase that frontier. Each protocell, each molecule that learns to copy itself, each mineral that catalyzes new chemistry, they are echoes of a time when matter first began to behave like life. And in those echoes, we can still hear the faintest whisper of the planet's first breath. Even with all the discoveries of the past century, one question still hovers above the field of origin of life research. A question that is as much philosophical as it is scientific. When did chemistry become biology? We can recreate amino acids, build lipid membranes, even make RNA molecules that copy themselves. We can simulate the oceans, the vents, the atmosphere of early Earth. But somewhere between those experiments and the first living cell lies a gap, a step that no one has yet seen happen. That gap is often called the emergence problem, the moment when non-living matter crossed a threshold and began to evolve, adapt, and persist. It sounds simple in words, but it's one of the hardest problems in all of science. Because the transition from chemistry to biology wasn't a single event. It was likely a slow drift, a long continuum of molecular systems, most of which failed, some of which endured just a little longer. At what point in that gradual process does it make sense to call something alive? If we could travel back 4 billion years and look into the warm oceans or deep-sea vents, we might not even recognize the earliest life. It probably wasn't a cell in any familiar sense. No nucleus, no DNA, no stable shape. More likely, it was a loose network of molecules performing reactions inside a fragile membrane, borrowing energy from minerals or heat. To us, it would look like a strange chemical froth. But within that froth, feedback loops were forming, small systems that made conditions favorable for their own persistence. That's where the line between living and non-living starts to blur. Scientists can describe every ingredient involved, but what's still missing is organization. Life isn't just complex chemistry, it's chemistry that maintains itself. A living system constantly repairs, reproduces, and adapts to change. It's dynamic stability, order built on motion. How that kind of self-sustaining organization first appeared remains unsolved. Part of the challenge is that the evidence is gone. The earliest forms of life left no fossils, no direct traces in the rock record. The first billion years of Earth's history have been erased by tectonics and erosion. We have only the faintest isotopic signatures, subtle chemical ratios in ancient minerals that hint that life might have existed as early as 3.8 billion years ago. But the actual molecular structures that started it all are lost to time. That makes recreating life's origin in the lab an exercise in reverse engineering without a blueprint. Scientists can test pieces of the puzzle, but not the whole system at once. They can build protocells that grow and divide, or RNA that replicates, or peptides that fold and catalyze reactions. But bringing all those elements together into a single, self-sustaining entity has proved almost impossible. The reason is partly scale. Early Earth offered billions of years and an entire planet as a laboratory. Our experiments have hours, weeks, or months, and a few glass flasks. Processes that once unfolded across oceans and volcanic landscapes must now happen in a few cubic centimeters of liquid. Even with modern technology, that's an enormous difference. Another difficulty is that life's chemistry is incredibly interdependent. In a living cell, every process depends on many others. Metabolism supports replication. Replication supports membrane maintenance. Membranes regulate energy flow. It's a network that only works once it's already assembled. A kind of chicken and egg paradox at the molecular level. Rebuilding that from scratch means finding conditions where each part can emerge gradually and remain stable along the way. There's also the problem of chance. The combinations of molecules that might lead to life are practically infinite. Most random chemistries don't do anything interesting. They just fall apart. Finding the rare ones that organize into feedback loops takes patience, creativity, and, increasingly, computing power. Some of the newest work uses machine learning to map chemical reaction networks, searching for combinations that spontaneously produce complexity. It's like looking for the path of least resistance through a landscape of nearly endless possibilities. Despite these challenges, progress is accelerating. Synthetic biologists are now constructing systems that exist in the gray zone between life and non-life, sometimes called proto-life. These are chemical assemblies that can reproduce, evolve, or metabolize, but not yet all three at once. They behave almost like living organisms, but remain fundamentally synthetic. At the J. Craig Venter Institute, researchers built the first minimal cell, stripping a bacterium down to just the genes absolutely necessary for survival, about 473 of them. By removing and reintroducing genes, they can watch how life's essential processes fit together, identifying the minimum requirements for something to be considered alive. In a sense, they are peeling back the layers of evolution, revealing what the earliest cells might have looked like. Other teams, like that of Kate Adamala and Pierluigi Luisi, have created artificial cells using simple lipids and RNA or DNA templates. These protocells can synthesize small proteins inside their membranes and even exchange molecules with their surroundings. They don't reproduce independently, but they show that the chemistry of life can be reassembled piece by piece. Meanwhile, researchers studying autocatalytic networks, chemical systems that promote their own growth, are finding examples that behave surprisingly like ecosystems. Certain mixtures of molecules can stabilize each other's reactions, forming tiny self-sustaining communities that compete for resources in the same way populations of living organisms do. These chemical ecologies might be modern analogues of prebiotic systems that once existed before biology fully took over. The ultimate goal is to create a system that can sustain itself indefinitely, a laboratory origin of life. No one has achieved it yet, but each generation of experiments gets closer. Some scientists believe we'll see a self-replicating, evolving protocell within the next few decades. When that happens, it won't just prove that life can emerge naturally, it will show that life's emergence is inevitable under the right conditions. But even if we succeed, it may never feel like a full answer. Because understanding the origin of life isn't just about the mechanics of molecules, it's about the nature of existence itself. Why does matter organize? Why do certain systems resist decay, adapt, and persist? Those are questions that reach beyond chemistry, into physics, philosophy, and perhaps something deeper about the universe's tendency toward complexity. For now, the frontier of synthetic life continues to blur the line between what is made and what is born. Each protocell that divides, each molecule that learns to replicate, pushes that line a little further. And maybe that's the real lesson, that life isn't a single event frozen in the past, but a process still unfolding wherever the conditions allow. The great unknowns remain, but they are shrinking. We may never pinpoint the exact moment when chemistry became biology, but we can now watch the same story play out again, molecule by molecule, in our own laboratories. And in that slow rediscovery, we are not just studying life's origin, we are participating in it, helping the universe remember how it first learned to live. Once scientists realized how early life may have taken hold on Earth, a natural question followed. If life began here so soon after the planet became habitable, could it happen elsewhere too? The more we learn about our solar system, the more that question shifts from if to where. For decades, Mars has been at the center of that search. It's our nearest planetary neighbor, and among the most Earth-like worlds we know, cold and desolate now, but once far wetter and more dynamic. Ancient river valleys, lake beds, and delta formations mark its surface like the scars of a lost ocean world. Rover missions such as Curiosity and Perseverance have confirmed that billions of years ago, Mars had liquid water, a thicker atmosphere, and the chemical ingredients needed for life. Rock samples from Martian craters show clays and minerals that can only form in water. The rovers have also detected organic molecules, compounds containing carbon and hydrogen, preserved in ancient sediments. These aren't proof of biology, but they show that Mars once hosted the same basic chemistry that existed on early Earth. In 1996, a meteorite found in Antarctica, known as ALH 84001, sparked enormous debate. It originated from Mars, blasted into space by an impact millions of years ago, and eventually landing on Earth. Inside the rock, scientists found tiny structures resembling fossilized microbes, along with magnetic crystals that some interpreted as potential biological products. The discovery briefly ignited hopes that we had found evidence of Martian life, but later analyses suggested non-biological explanations. Even so, the controversy marked a turning point. It made the search for life on Mars a legitimate scientific pursuit, not just a speculative one. Since then, new data have made the planet even more intriguing. Mars orbiters have detected seasonal methane emissions, plumes of gas that rise and fall with the temperature. Methane can be produced geologically through reactions between water and rock, but on Earth it's also made by microorganisms called methanogens. If something similar existed on Mars, even deep underground, it might still be there today. Future missions aim to find out. Perseverance is currently caching samples for return to Earth, where scientists will analyze them for chemical signatures that could reveal whether life once existed on the red planet. But Mars isn't the only world where life might hide. As our technology has improved, attention has shifted to more distant and surprising places, the icy moons of the outer solar system. In 1979, NASA's Voyager spacecraft flew past Jupiter's moon Europa, and what it saw stunned scientists. Beneath its bright, cracked ice surface appeared to be a vast subsurface ocean, kept liquid by tidal heating, the gravitational flexing caused by Jupiter's pull. Later missions, including the Galileo orbiter, confirmed the evidence. Europa likely contains more liquid water beneath its crust than all of Earth's oceans combined. That discovery changed everything. It meant that life might not need a warm, sunlit surface. It could exist in the dark, heated from below, just as microbes thrive around hydrothermal vents on Earth. In 2013, the Hubble Space Telescope detected water vapor plumes erupting from Europa's surface, jets shooting hundreds of kilometers into space. If those plumes connect to the ocean below, they could carry traces of whatever chemistry is happening down there. NASA's upcoming Europa Clipper mission, launching soon, will fly repeatedly through those plumes, sampling them for organic molecules, salts, and possible biosignatures. It's a search for what scientists call habitability, not proof of life itself, but evidence that life could exist. Another moon, Enceladus, a small icy world orbiting Saturn, has proven even more dramatic. When the Cassini spacecraft passed near it in 2005, it discovered enormous geysers erupting from fissures near the moon's south pole. These plumes contained water vapor, methane, and complex organic compounds, all venting from a subsurface ocean beneath the ice. Cassini's instruments also detected tiny silica particles, the kind that form only in hot, mineral-rich water. That suggests the ocean floor of Enceladus is home to hydrothermal activity, conditions strikingly similar to the deep-sea vents where life on Earth may have begun. In 2017, scientists analyzing Cassini's data found evidence of molecular hydrogen, H2, in the plumes, a potential energy source for microbes. On Earth, hydrogen and carbon dioxide power the metabolisms of some of the oldest life forms we know, including methanogens. The same chemistry could, in theory, sustain a microbial biosphere inside Enceladus's hidden ocean. It's difficult to overstate how profound that possibility is. In a solar system once thought barren, two moons, Europa and Enceladus, may each hold global oceans with the right conditions for life. Neither depends on sunlight. Both are heated from within and rich in organic materials. If life could arise there, it would mean that biology might be a natural consequence of planetary chemistry, not a rare accident. Beyond these icy moons lies another world that challenges our ideas about habitability, Titan, Saturn's largest moon. Titan has lakes, rivers, and rainfall, but not of water. Its liquid is methane and ethane, flowing under an orange sky filled with organic haze. At minus 179 degrees Celsius, minus 290 degrees Fahrenheit, it's far too cold for liquid water. Yet Titan's atmosphere is a rich chemical factory, constantly bombarded by ultraviolet light from the sun and by cosmic rays. These energy sources drive reactions that build complex hydrocarbons, long chains of carbon and hydrogen that on Earth form the backbone of organic chemistry. When the Cassini-Huygens mission landed on Titan's surface in 2005, its instruments detected tholins, reddish organic particles formed by atmospheric chemistry, and a landscape that looked eerily familiar: river channels, deltas, and shorelines. Titan is often called a laboratory for prebiotic chemistry, a place where we can study how complex organics behave in a completely different environment. It may not host life as we know it, but it could show us how life could begin in another form. And even beyond our solar system, the building blocks of biology appear to be everywhere. Astronomers have detected amino acids, alcohols, and even sugars in interstellar clouds, some thousands of light-years away. In 2019, scientists found glycolaldehyde, a simple sugar, in a star-forming region of space. Radio telescopes routinely detect formamide, a molecule thought to be crucial for forming nucleic acids like RNA. Comets, meteorites, and dust grains throughout the galaxy are loaded with organic molecules. In a sense, the universe is constantly performing the chemistry of life, scattering its products across space. This abundance raises a tantalizing concept: the idea of a second Genesis. If life arose on Earth and then independently on another world, Mars, Europa, Enceladus, or anywhere else, that would prove that life isn't a cosmic fluke. It would mean that biology emerges wherever conditions allow. Finding even microbial life with a completely different biochemical structure, using different amino acids or a different genetic code, would be one of the most important discoveries in history. Such a finding would answer a question that has lingered for centuries. Is life universal or unique? If it's universal, then the cosmos is likely filled with living worlds, each shaped by its own environment, but built from the same elemental logic. Life might not be the exception in the universe. It might be the rule. Even if we never find a second Genesis in our solar system, the search continues across thousands of exoplanets orbiting distant stars. Modern telescopes like the James Webb Space Telescope can now analyze exoplanet atmospheres, looking for chemical imbalances, the fingerprints of life. Gases like oxygen, methane, and carbon dioxide in the right ratios could indicate biological activity. Already, Webb has detected signs of carbon-bearing molecules in multiple exoplanet atmospheres, and future missions will push that sensitivity even further. What would it mean if we eventually confirm life beyond Earth? It would redefine our place in the universe. It would suggest that life is not the outcome of chance, but of physics, that when the right elements meet under the right conditions, life is the natural continuation of chemistry. It would tell us that the cosmos itself is fertile, that every planet and moon is a potential experiment in becoming alive. But even if we find nothing, no microbes on Mars, no biosignatures on Europa, the search still matters, because the act of looking teaches us what makes Earth's life possible. It reminds us how rare and fragile our biosphere is, and how small the window of habitability might be. It brings perspective that everything living here, from the simplest cell to the most complex brain, is part of a cosmic continuum stretching back 13.8 billion years. Life's potential beyond Earth is not just a scientific question, it's a philosophical one. It asks whether the universe produces consciousness as naturally as it produces stars, or whether we are a solitary accident, a brief spark in an otherwise indifferent cosmos. Either answer is profound. If life is everywhere, then the universe is full of mirrors reflecting our existence. If life is unique to Earth, then this planet carries something infinitely precious, a phenomenon found nowhere else. For now, we keep searching, beneath Martian dust, within the icy shells of distant moons, in the faint chemical whispers of faraway atmospheres. And as we do, we're not just hunting for alien microbes or fossils. We're searching for connection, for evidence that the story that began here has also begun elsewhere. Because if we find that life has sparked twice, even in the same solar system, it would tell us something fundamental about the nature of reality. That the universe doesn't just allow life, it wants it. If you trace the story of life far enough back, the trail fades into chemistry, and then into the quiet physics of a young universe. Every atom in your body, every thought in your mind, began as something simple and indifferent. Hydrogen born from the Big Bang, carbon forged in a dying star, oxygen scattered by a supernova. From that chaos came order. From energy, structure. From structure, complexity. And eventually, consciousness. It's difficult to fully grasp how improbable that chain is. Over billions of years, the universe assembled dust into planets, planets into oceans, oceans into molecules, and molecules into something that could notice it existed. Matter learned to think. The same elements that once drifted through interstellar clouds now sit behind eyes that look back at the stars, asking how it all began. That is perhaps the most astonishing outcome of evolution, not just that life appeared, but that life became aware of itself. Somewhere along the way, carbon and water began to dream. The Earth was never required to produce life. For most of cosmic history, it didn't. It could have remained just another rock orbiting a star, silent and unremarkable. But through an extraordinary sequence of conditions, a stable orbit, a magnetic field, water, chemistry, time, it crossed the threshold into biology. And once that happened, life adapted to everything the planet offered. It learned to breathe, to photosynthesize, to move, to feel, to think, and eventually to wonder. We don't know whether this story is unique to Earth or repeated endlessly across the galaxy. But even if we are alone, that only deepens the miracle. Because the rarity of life doesn't make it less meaningful. It makes it immeasurably precious. The fragile biosphere that surrounds this planet, the thin film of air, water, and warmth, is the only known cradle for consciousness in the universe. And every living thing, from bacteria to humans, is a continuation of the same ancient thread that began in a quiet ocean 4 billion years ago. When you breathe, you are participating in that lineage. When you think, the atoms of stars are reorganizing themselves into awareness. You are a temporary arrangement of ancient matter, one that remembers, plans, creates, and dreams. That's what makes life not just a scientific mystery, but a philosophical one. The universe has given rise to something that can contemplate its own origin. And when you look up at the night sky, at those billions of silent, distant worlds, you're seeing the same cosmos that once stirred the first spark of life here. It hasn't changed much. The same physical laws that govern galaxies also govern the molecules in your cells. The same starlight that forged the chemistry of early Earth still shines down today. So perhaps the story of life isn't about luck, but inevitability. Given enough time, energy, and possibility, the universe seems to find ways to organize itself into something that feels, perceives, and asks questions. And in that sense, you, listening now, thinking now, are the latest expression of that original curiosity. The first cell became you, and you are still part of that beginning. From chaos to chemistry, from molecules to minds, the line is unbroken. And as long as life endures, the universe will keep reflecting on itself through the fragile, brilliant phenomenon we call being alive. So wherever you are, take a breath and remember, the same breath once moved through oceans that had no life at all. Now it moves through you, the living continuation of the first spark that ever learned to live. Thank you for listening. If you enjoyed this journey, take a moment to subscribe and share where you're listening from. It helps this channel grow and keeps these stories alive. Wherever you are, I hope this left you with a sense of wonder, that life, fragile, improbable, and beautiful, continues to unfold around us every moment. Until next time, sleep well and keep wondering.